on the last slide. So with that epithelial cell barrier, you've got, and especially in the skin, you've got your outer layer that forms the barrier itself, and that are some level of differentiated cells. For the gut, for the lung, you've got that one layer of terminally differentiated epithelial cells. In the skin, in the genitourinary tract, you've got several layers that those cells are in various stages of differentiation. Okay? And then you've got that basal cell layer, which is the stem cell layer, and it's typically only a single cell thick, that are your undifferentiated stem cells, that then when the cells are sloughed off and they need to be replaced, those stem cells then begin differentiating and produce the terminally differentiated exterior cells. Okay, you've got that underlying level of connective tissue, so those are the three levels that make up that epithelial barrier. The epithelial cells proper, and then that supporting tissue under it, where there's a lot of really important pieces that go into the function of that epithelial barrier, because that's where a lot of the immune cells are located, those dedicated lymphoid follicles, whether it's the Peyer's patches in the gut, or the other organized structures in other epithelial barriers. All right? But then also, you've got dedicated sets of lymph nodes that are located near those structures. So in the gut, they're the mesenteric lymph nodes. And so there's sort of a set of lymph nodes that are, in close proximity to the GI tract. In the lung, you've got the hilar and the mediastinal lymph nodes that are located here around the heart, so sort of midway in your lungs, right? And so those dendritic cells, those macrophages that become activated at the epithelial barrier, don't have very far to traffic, and vice versa. The T cells don't have very far to traffic to then get to the infected cell surface. So it's efficiency of the system that of the immune response coming into play. All right? And then your basic function of that epithelial barrier is to be a barrier, to protect against infection, to protect against damage, but then also major role is being able to differentiate that normal, basic, good, healthy commensal flora that helps our immune response versus the pathogenic organisms. All right. We've talked about all of the major epithelial barriers. There's a couple more. Your eyes, ocular is actually considered a mucosal barrier, completely different. And then we've talked about the other three in the last hour. And at each of these types of epithelium, they're very specialized, but there are some common features. So there's that continual turnover of cells, especially this time of year, shedding all of that dry skin. That works in many ways. Number one, it gets rid of dead cells, but at the same time, it's getting rid of a lot of that bacteria. So at a time where there's a lot of infection going on, thinking about flu right now, or if you've traveled to the wrong place, another virus, that can bind to that, and then you can shed those infected cells potentially. And then those cells have to be replaced to generate that good protective barrier, and so there you have the stem cells, that one basal cell layer of the cells that then get the signals to differentiate and then repopulate, replace those shed cells. Okay? As those stem cells differentiate, they move toward the surface. Where it will become important in talking about the skin is because there's different stages of differentiation, a process as those cells push up to that final outer layer of the keratinized epithelial cell. These cells are immunoprivileged, all right? We kind of touched on that term. Immune response, privileged, protected. So there are different mechanisms in place that keep the immune response from targeting the epithelial cell itself in a normal state, right? So lots of Tregs, lots of specific proteins that are expressed by those epithelial cells that says, don't touch me unless I send out the big SOS or red flag, okay? So that's how we have some of these mechanisms, like the homing mechanisms, the vitamin A with the GI tract. We'll talk about vitamin D in skin here in a few slides, that then really send out that big signal saying, all right, I'm in trouble, come and protect me. all right, as an epithelial barrier, all right? Okay, and again, specialized structures, same thing as we talked about in some specifics last hour. You've got a mix of cells, you've got a mix of specialized secreted proteins that all function in a protective manner, okay? You've got either the single layer or several layers. We didn't really talk about columnar versus cuboidal. You don't need to really think about that. Maybe thinking about the ciliated versus the non-ciliated. What we didn't talk about in the respiratory epithelium is the function of cilia. So if you look at a respiratory and a GI epithelial cell, you'll see these extensions. So in the GI tract, those microvilli, they're absorptive cells. So that's the job of that cell. It's absorbing the nutrients from the gut lumen. That's similar structure in a respiratory epithelial cell is called cilia. And its job is to brush. They're very modal. And so they take all the mucus, they take all of the particles, whether it's microbial or other, that get trapped in the mucus, and then brush them out of the respiratory tract. Because the other function of some of these epithelial barriers are other protective mechanisms. We're talking about chemical and biological, but there's also physical. So the physical sweeping of that mucus by the cilia, the process of sneezing and coughing, the process of peristalsis in the gut. It's forcing organisms, forcing those contents in the lumen of those structures out of the body as part of the, basically, just elimination of that potentially infectious organism, okay? Diarrhea is really nasty, but it's a good response because it rids the body very rapidly, very efficiently of whatever that infectious organism is. Same thing with a sneeze or a cough. That's why it is spreading it to everybody, but it's getting it out of your system. That's where your system becomes a little bit, or a whole lot, selfish, all right? It's protecting me, protecting this organism, not necessarily the community. And then we talked about the PRR expression. Okay, so the epithelial cells have to be able to differentiate because you've got the stem cell, and it must become that final terminally differentiated, fully functional. It must be tolerant to flora, but then be able to differentiate and respond to pathogenic organisms. And then you also have the unique sets of APCs at each barrier. We've talked about Kupffer cells in the lung, in the brain, even though we're going to talk about them, but just so that you know there's differences. In the brain, those specialized macrophages and dendritic cells are called microglia. In connective tissue, they're called histiocytes. And in the skin, you have two sets called Langerhans cells, and then dermal dendritic cells, which we just discovered about 10 years ago, are just differentiated. All right. So another specialized set of cells in epithelial barriers are those intraepithelial lymphoid cells. All right. So we're talking about three different sets here. So first are those innate lymphoid cells, then the conventional alpha beta T cells, and the gamma delta T cells. All right. Most of these are in some level of activation. So especially your gamma delta and your innate, your ILCs. But then your conventional T cells are typically in a memory state unless there's an active infectious process going on there. Don't need to know any of these numbers. If we're talking about the IELs, and so here, when we're talking about IELs, so this is a good example there. Okay, so there's not one on this slide. That gray cell, instead of reading melanocyte, pretend that's a gamma delta T cell, because that's where it would be living. It's at that epithelial barrier, or it's actually interdigitated into the epithelial barrier. It slips between two of those epithelial cells and then sends out different processes that are sampling the environment to say, all right, are you a phosphoantigen that I can recognize and then respond? So that's when to think about the word intraepithelial. It's inside the epithelial barrier, all right? And so the CD8 T cell, so since most of the gamma delta T cells are CD8 positive, You can also think about that purple cell on the far right as a gamma delta T cell. So a lot of them are in there. If we're looking in the epithelial barrier itself, most of the T cells are going to be gamma delta. If we're looking at the subepithelial, you've got a lot of your conventional T cells as well as some gamma delta T cells. Okay. So just as a recall on your gamma delta T cells, remember they're the non-conventional, leave the thymus very rapidly. They'll go all the way through the process. They can respond in both TCR dependent and TCR independent manner. So this is what kind of sets them apart from the conventional T cells. So they do recognize antigens through their T cell receptor. These tend to be phosphoproteins, recognizing maybe that phospho group on the molecule as far as the primary antigen. Their TCR independent activation occurs through the PRRs and especially the TLRs that they express. What do? They respond very rapidly. They respond almost in a timeline like an innate cell, but because it's an antigen-dependent process, because they express the CD3, it's an adaptive response. Okay? So they can produce cytokines, so they're immediately affecting differentiation of resident macrophages and dendritic cells in the area and T cells in the area. They're activating those macrophages and dendritic cells to function as APCs, and so they're giving them some of the signals to get out of the epithelial surface to the lymph node and make the response. And then they are directly cytotoxic. So if it's an antigen that they can kill through either the TCR MHC1 interaction or other ligand receptor interactions, they can cytotoxically damage and kill those target cells. All right, we've touched on ILC, so the innate lymphoid cell, a little bit, but we haven't maybe talked about them in much detail, so we've got a couple slides on that. So these are innate cells. They do not express CD3, they do not express TCR, they do not go through any of that lymphoid development that we talked about, so they are innate cells. However, when they function, they look a lot like a CD4 T helper cell. All right? What triggers their function? Responding to PAMPs and DAMPs through TLRs, and then most of them express a C-type lectin PRR. Right? So those two classes of PRRs. And then they have the different profiles. So see on this slide, you can see on the last slide as well. So just like that image that we had when we talked about CD4 T helper cell differentiation into TH1, TH2, TH17, we have the same type of responses occurring with the ILCs. Based on the PRR signal, other transcription factors are turned on in that cell that then cause a signature cytokine profile to be generated. So you have ILC1, ILC2, ILC ... however many there are at the moment, because of the differentiation and the cytokine set of cytokines that those cells express. Don't need to know what any of those are, but I would like you to appreciate you've got different specialized fine-tuning of the immune response, but different subsets of those cells that are then induced and differentiated to appropriately respond, to tailor that immune response to the exact pathogen that we need. Is it a virus? Is it an extracellular bacterium? Is it a parasite? An intracellular bacterium? All of these types of pathogens need a different type of immune response to clear it most effectively. And so we've got to fine-tune the response. And this is one of the earliest ways we have of doing that. All right? The ILCs, if you make enough, did I just lose? All right. Can you all hear me in the back without? All right, I'll change the battery while Dr. Palmieri is leading his slides. Okay, so if the cytokine response is robust enough that it can activate the macrophages, call in the neutrophils, then you can get efficient clearing of that invading response, of the invading microbe. So that's job number one, actually participating in a direct elimination of the invading organism. But maybe more importantly, it starts preparing the environment for the adaptive immune response that's going to come in, or for the amplified innate immune response. It's setting the cytokine, the right cytokine environment. So as soon as those cells hit that area of infection, they're bathed in the right cytokines to say, this is the way you need to respond. Okay? So again, it's all about the efficiency, because the immune response is programmed at many different levels to eliminate that pathogen, to repair that damage as quickly as possible for the good of the host. All right, then you've got all the commensal flora. We've talked about those many times. What I will say, so one of the advantages, you're out competing those invading microbes. Number one, just for space. There's a geography. It's all about the real estate. So if there's a lot of commensal normal flora setting there, then that invading microbe may not necessarily have room to attach and then cause the infection. They're also competing for nutrients. Yeah? If the nice commensal bacteria are using all the nutrients, then the invading organisms may be starved out. Right? So you've got competition for resources, and then because of the different factors we talked about in the last hour, you've got actual active defense mechanisms induced by the normal flora that protect against those pathogenic organisms. All right? And you've got a huge number of organisms. You've got at least 10 to 100-fold more bacteria in your body than you do all of the cells that make up your body itself. Vast number and vast diversity in those populations. Okay, we now call this our microbiome. That term was coined 10 or 12 years ago. And you can read about that. None of this would be testable, but really interesting statistics that may be a little bit out of date, this is 5 or 6 years old, so haven't seen a newer version, but there it is. And then this gives you an idea of just the density and the competition that hopefully people partake. All right. So how do we get this microbiome? We're inoculated at birth. There is a dramatic difference in the microbiome for babies who are born vaginally versus babies who are born by C-section. So part of that process of traveling through the birth canal is inhaling, sampling mom's flora to start that colonization in you as a baby. And then you've got that great diversity that changes throughout age, with geography, with all of the different exposures, whether that exposure is food versus drugs versus just the environment. Right? Here's one example. So you're going to see several images like this. Do not at all pay any attention to what all of those different colors mean. They all are a different organism. So this is just looking at the fungi on your body. And so if you look at the colon versus the lung, because we talked about the GI tract, versus the oral cavity, so those are the 3 mucosal surfaces we talked about last hour, just look at the differences in the color distribution. Those are all the differences in the fungal microbiome, and this is just fungus, and that's a minor part of our microbiome. Those are all the differences just at sites in a single individual. So again, that specialization or that difference in exposure and environmental conditions. All right, we talked about cytokines and defensins, so I'll leave you to kind of remember that if you want to read it again. That's the same thing that we talked about in the last hour a little bit. Specialized mediators, we talked about the role of IL-17. One that is new, that it plays a more important role in the epithelium, in the skin, the cutaneous epithelial defense is TSLP, so thymic stromal lymphocyte. And so that is it's produced by the cutaneous epithelium and some fibroblasts there. It's a strong activator of dendritic cells, and then it tends to induce type 2 T cell responses versus type 1. All right, and again, just kind of summarizing, but comparing the 3 major epithelial cell barriers that we're going to talk about in last hour and this hour. Cutaneous, skin. Okay. All right, so cutaneous epithelium, those cells may also be called keratinocytes. different layers from the undifferentiated cells here in your basal keratinocytes, and then as they progress through the differentiation process, they reach that final stage and become stratified, and that's just they become very hardened, they're dead at that point, but that becomes the more impenetrable barrier that is the skin, and so you've got different levels of differentiation through this, but that outer layer is then called the stratum corneum, right? Then under that, you have your dermal layer, and then within all of these, you have very diverse cell populations. All right, so you've got a different set of Velcro molecules here. So for your GI and respiratory epithelium, your connections are specific sets of integrins that we mentioned in the last hour when we were talking about the trafficking. So you have other integrins, other cell adhesion molecules that hold those epithelial barriers together in the GI and respiratory tract. For the skin, you have some specialized molecules called connexins and filaggrin. So connexins are actually a family of proteins. Don't really need to know any of the details about those, other than to know that as the epithelial cell layer is differentiating, you have different connexins that are expressed. So just different molecules, different level of adhesion. And so when you see the CX, whatever, CX is connexin. So just what I would hope you remember are connexins or adhesion molecules, a family of adhesion molecules for epithelial keratinocytes. All right. Then we have filagrins. And so filagrins, filament aggregating protein. So filaggrin is produced as a profilagrin that is then digested and processed into the final filaggrin state. So these are basically ropes of protein, filaggrin monomers that then are roped together to form that strand of filaggrin that binds keratin as it is being produced in the outer layers, some of the final steps of the differentiation process. And the filaggrin and the keratin form this mesh that holds all of those epithelial cell layers together. Okay, so a filament process. Okay. Why this is really important is because we have specific mutations that we know contribute to autoimmune or at least immune-mediated dermal diseases. And Dr. Prater will talk to you about some of those. They're listed here, but you don't need to know those. For me, right now, you've got a lecture of immune-mediated dermal diseases or cutaneous diseases where you'll talk about the specifics there. But this is why maybe you need to know a little more detail about filagrins. All right? And here you can see all of these strands. Those are the filaggrin monomers that are forming to bonding together with keratin to make that mesh. All right. So keratinocytes. Remember, keratinocytes are the cutaneous epithelial cells. They secrete lots of factors. They're secreting those connexins and filagrins. They're secreting a lot of cytokines, things we've talked about multiple times before, but there are some of the three big players as far as the three major functions that those keratinocyte-derived cytokines need to perform. All right, so what are some of the specialized cells? Number one, you have the specialized dendritic cells, the Langerhans of the dermal cells. Then you have the ILCs, you have the specialized T cells. Here we're really thinking about the gamma delta T cells, specialized because they respond so rapidly and respond in a different way from our conventional T cells. This mix of cells are organized. So what you have in the epidermal layer versus the dermal layer is different. In the epidermis, you have the Langerhans cells, some memory conventional CD8s, and then lots of those gamma delta CD8 cells. Whereas in the dermis, here in the lower part, you have the dermal dendritic cells and more CD4 cells, memory cells, and some CD8, some gamma delta, and a lot of ILCs. Right? So there's, the separation of powers, if you will. But again, it's that specialization of response. All right. Okay. So how do we know that there are different sets of dendritic cells? They look different. They're phenotypically different. You all haven't had the immune assay lecture yet. For those of you who don't know, this is a flow cytometry dot plot. How does this work real quickly? You stain the cells with different antibodies. This is why we're teaching you all of those CD numbers. And so if you look here on the x-axis, this is staining for CD1. Doesn't matter what it is, but it's staining for one set of proteins or one specific protein. And then on the y-axis, you're staining for a second protein. Each of those little dots is a single cell. So this, once you stain the cells and they have a fluorescent tag on them, the cells are then put through a machine, a flow cytometer, that has a laser that activates those fluorescent labels, those fluorochromes. They all give off, they emit light at different frequencies, different wavelengths, and then you have detectors that can detect each of those different wavelengths and say, okay, I see one cell or 10,000 cells that are expressing, that have this fluorochrome bound to them, where there's a second detector that will detect the fluorochrome attached to that guy. Everybody kind of understand that process? Okay. So, just so you can see, here are your dermal dendritic cells that are expressing very high levels of CD1. Again, you don't need to know the CDs, but expressing very high levels of CD1, but absolutely no CD14. Versus your dermal, so these are Langerhans cells. So the paper just called them epidermal, but if you go back to the last slide, Langerhans. Then you have your dermal DCs, which are expressing very high levels of CD14, that's the upper box, but very low levels of CD1. So that's a way that you can differentiate different cell populations in whatever. This is the way they phenotype cancers, lymphomas, leukemias, by staining for these different patterns of markers, okay? This is how we can differentiate and say we know there are two different sets of dendritic cells in the skin, right? So you've got different cells, you've got different locations, one in the epidermal, one in the subdermal layer, and then you've got different functions because of the trafficking patterns and the types of cytokines that each of these cells tend to make. Langerhans cells are biased for making type 1 T cell responses. The dermal dendritic cells are biased for making type 2. Okay? We've talked about gamma delta T cells a couple times, so we'll skip this slide. Okay, detect, that's on a few of these slides. I have defined it here just so everybody is not asking what detect stands for or what it is. You do not need to know this. There's a slide, it's 42, it's a supplemental slide. If you want to know a little more about dermal epithelial T cells, they're very specialized T cells, but you guys will not be tested on that. But I just didn't want you to see something repeatedly on these slides and these figures that might create a lot of questions. All right? Okay, so what happens when you have these compromised barriers? The insult can be physical, trauma, that cut. It can be chemical. UV is a chemical signal. That sunburn is chemical damage to the skin. Or it can be biologic. You have altered integrity. You have altered permeability. Things can get in. That barrier is changed. One of the first responses is going to be making lots of cytokines, causing the differentiation of the keratinocytes to replace the damaged cell, recruiting in the right immune cells and setting the right cytokine mood environment for those recruited cells to be able to differentiate into the right way. Sources of cytokines in the skin, you've got the keratinocytes, you've got those APCs and the Langerhans and the dermal dendritic cells, you've got some effector CD4 T cells, your conventional cells, and then not listed on here, which really should be, is your ILCs, so those innate lymphoid cells that look, that act a lot like a T helper cell, but are not T cells. Remember, they're innate cells. But the key point here is setting up the right cytokine environment so that you have trafficking of the dendritic cells to the lymph node to educate the T cells, activate the B cells, and then bring them back, the T cells and the antibodies, to that infected site in the skin. Okay? You can have a few naive and effector cells present, T cells, in the skin, but they're going to be transient. predominant presence of conventional alpha-beta T cells are going to be a memory cell of some type. All right, just remember that. Yes, I put in the burgundy that there are different, so you've got CD4 TEM versus RM, and then CD8 RM. Don't know those specific titles. If you remember from our T cell lecture, we said that there were different types of memory cells. We don't understand how we are. There are 3 different models about how different memory cells are generated. That's all you need to know, and you need to know that you've got a mix of different memory cells, but as far as the predominant conventional T cells in the skin, they have a memory phenotype versus an effector phenotype, as far as that long term, not the transient. Does that make sense? Okay. All right. So, how do those T cells find their way back to the skin? We've got another homing mechanism. This is why we are always hearing, well, go out and get some sun and soak up that vitamin D. It's also you can protect your epithelial, your cutaneous immune response. Because in the lymph nodes, those epithelial APCs are responsive to vitamin D-induced signals. All right, so UVB irradiation triggers vitamin D synthesis in those dermal epidermal dendritic cells, in the Langerhans cells, and in the dermal dendritic cells, and in the other macrophages that are and dendritic cells that are resident there in the skin. When that cell, the APC, becomes activated, as it starts to traffic back to the lymph node, or traffic to the lymph node, it converts that pre-vitamin D, it takes up the pre-vitamin D that's in the environment, and then converts it to its active form, right, of full vitamin D. When that APC then presents the antigen to the vitamin D, to the T cell, to the naive T cell, it releases that vitamin D, and that vitamin D imprints a signal on that T cell that says, you need to go back to the skin, just like vitamin A did in the gut epithelial response, right? And then we're getting expression of specific chemokines, expression of specific integrins based on that vitamin D-induced signal in the T cell that then causes the trafficking, causes the T cell to leave the lymph node, enter the circulation, and then go back to the skin, right? Same process, basically, 2 different vitamins based on which epithelial barrier we're talking about. All right. And so, again, microbiome is hugely important. And, anybody heard personalized medicine? Yeah, okay. The microbiome and manipulating the microbiome is a huge part of that process, or that concept. And so, just looking at a single individual, so imagine volunteering for this study. You went in and you had every single possible skin surface swab. All right, yeah, okay. So this graph now looks at 4 healthy volunteers. All right, so here, HV1 through 4, those are our healthy volunteers. And then they're showing just 4 different sites. So the back, the nose, the heel, all right. And again, look at the differences in colors. Number 1, just comparing, if you look at the first bar in each set of anatomical locations, that's the same volunteer. That's all volunteer number 1. So even on his own body, he's got very different colonies, diversities of those microbes at those different anatomical locations. All right, but is it the same mix? So if you look at the nose of every single person, is it the same? No, they've got some of the same base, but different balances, and then there's going to be unique organisms there. All right? So you've got great diversity in the response between sites, or not in the response, but in the colonization, between sites and between people. All right, so what does that mean? You've got the innate immune responses that can be triggered, and you've got the production of those antimicrobial molecules, because you're getting that low-level tickle of the immune response that says, talked about how the commensal microbiome can do that. We've talked about that dysregulated cytokine production that can happen when the microbiome becomes dysregulated, when there's that balance that is disturbed between the commensal versus the pathogenic, or when there's another change in the body and those commensal organisms now become pathogens. Somehow that level of, and again, I'm going to use this word loosely, because the next lecture is tolerance, and you'll see why I've sort of hedged when I say that tolerance to the commensal organisms are broken. So it's not an immunological tolerance, so be very careful about using that word maybe, but that functional tolerance, saying these are bacteria, yes, but they belong, they're helping me, so I'm not going to mount a response to them. If the metabolic, if the biochemical balance in the body can become dysregulated, we can begin to make a response to those commensal organisms. This is the case in some of the autoimmune diseases, like psoriasis. There's not any, well, there are some mutations, but there's not a pathogenic organism that triggers that. This is all being triggered by a dysregulated immune response to normal commensal bacteria. And then you can have the introduction of those pathogens that they raise the immune response to that threshold, cause that inflammatory response, but then that can exacerbate, that can worsen existing conditions because you've got that dysregulated immune response that can alter the normal, well, not the normal, but the disease condition. All right, here are some examples of that, and again, this is not testable because you've got a whole lecture on immune-mediated skin diseases, but we know that this happens. Hypersensitivity. You know, you've got that bee sting. You put on that piece of jewelry that has a lot of nickel in it, maybe, and all of a sudden you're itchy and your earlobe is red and swollen and maybe starting to do stuff. That's that hypersensitivity response. IgE, mast cells, leukotrienes, all of these things that are being triggered. Psoriasis, atopic dermatitis. So immune-mediated diseases in the skin because of those inappropriate responses to commensal organisms, whatever the underlying mechanism is. Skin cancer, right? UV damage, that activates a lot of pro-inflammatory cytokines. It damages the DNA, but it also induces some immunosuppressive, so you're suppressing the immune response so you can't repair and fix the damage caused by that UV-induced DNA damage, and so now you've got a metastatic event and you have skin cancer. So dysregulated immune response resulting in disease in the skin. Okay, so none of the rest of this is testable, but this is where the future of epithelial cutaneous immunity is headed. Some of that personalized medicine and some things that maybe are being done in one of the buildings here in the Corporate Research Center. All right, because we can now look at a lot of the events that are going on in the skin. We have the tools that we can phenotype cells, like we've seen with the dendritic cells. We know how we can maybe manipulate those in the lab. Why is this important? So let's just think about wound healing, all right? So a lot of head and neck surgery fails because the skin, you think about how mobile, especially some of us, you know, your head and your neck is, your facial expression. So if you have surgery here, one of the major problems with that failure is because you don't have good healing because of the continued movement. So you can't maintain that integrity of those newly forming epithelial cells long enough for all of the connections to be really knit in place. All right, skin. You know, if you want to read about that, you know, Google skin flap surgeries. Okay, this was a area of research that we were just starting at the biotech company I worked in Maryland before it shut down. But how can we accelerate that healing process for those skin flap surgeries? You know, all the head and neck cancers that we're talking about now. Or think about burn patients. Their major mechanism of mortality, their levels of mortality, is because they lose so much of the skin, they're not protected against infection, and it takes so long for that skin to form can we now manipulate in a good way the immune response or all of the cytokines? Remember all those growth factors we talked about? Some of those are needed to make skin. Epidermal growth factor is a cytokine that promotes differentiation and generation of epidermal skin cells. Okay, so how can we use science to now accelerate that medical process or help those medical processes? Food allergies in the skin, we talked about in the last hour. Is our inappropriate, maybe, delivery of these food allergens on the skin of those babies now resulting in food allergies when they start eating that food because their skin cells have been taught that this is what we need to tolerate to that coconut oil moisturizer or whatever it is. But then when that food is consumed, because it's already tolerized here on the skin, the immune response now says, all right, this should not be in the gut. It's supposed to be on the skin, and begins to make that immune response. Is that the mechanism? Or is there some way that we can, because we know there's a lot of tolerance that can be induced, is there some way that we can find whatever those food allergens are and find a way to now induce tolerance so that celiac patient can eat a regular pizza crust or donut or whatever? All right. Interdermal vaccines. Again, we haven't had the vaccine lecture yet, but how are most of our vaccines given? In the skin? No. They're given in the muscle, maybe subcutaneous. For those of you who've looked at the syllabus, is there a muscle that, is there a lecture that's entitled Immune Responses in the Muscle? No. There's very few cells there. There's no dendritic cells, I mean anything except transient just because you've got blood flowing through. But the muscle cell doesn't have a lot of immune cells there. T cells, whatever. They're not there. There's none of these resident cells. So do you think giving a shot in the muscle may be the most efficient way of delivering a vaccine? Not really. But that's how we're giving them. It's nice, it's easy. One of the most effective vaccines we have, smallpox, completely eliminated it, is given in the skin. All right? Think about all of these dendritic cells. If you go back 10, 12, 20 slides to where I had that immunofluorescent that had the Langerhans cells and the dermal DT stayed fluorescently, there's a lot of dendritic cells in the skin. We know from history that that vaccine strategy works. So how can we now maybe develop vaccines that we can deliver in the skin that may be more efficient? Maybe we can take it from 1 or 2 doses, from 4 or 5 doses with all the boost down to 1 or 2. A single dose of smallpox vaccine theoretically gives lifelong protection. Most people are only, when we would immunize them, are only immunized once. Okay? Only on certain conditions are people boosted every 10 years. If you work with vaccinia virus like I do, or if you're a first responder, just so you are insured if you're going to be put in a place of danger, just to be sure you've got that good immune response, you get boosted. But for those of us who were old enough to have been immunized, in the U.S. they stopped immunizing in the mid-70s. That can give you a little idea maybe of where I am in the lifespan of things. But all of those people, my parents, probably most of your parents, only got 1 shot of the smallpox vaccine, and they're protected. And then as we now understand more about dendritic cells, more about these cell populations, not just location, making the vaccines intradermal, but how can we formulate that vaccine so that we're inducing the right type of immune response, whether it's at the dendritic cell level, activating the right set of ILCs to differentiate, or just setting up that correct immunological cytokine environment for the cells to differentiate into.